going. Then share this PowerPoint. Oh, I didn't put up Friday's lecture. My bad. Yeah, I'll make a note of that and do that. I think it might have been. Yeah, it's very possible I didn't upload it. But yeah, I'll make sure to do that. Thanks for letting me know. All right. So what we were talking about on Friday. Yes, I will upload the Friday lecture. Yep. What we were talking about on Friday was we were talking about secondary structure. And just as a reminder, secondary structure is just the local conformation of our protein. And we're basically talking about alpha helixes and beta sheets. We talked about the backbone, how we have fine psi angles, and how we can calculate a dihedral angle. And then we talked about alpha helixes, and we talked about proline and glycine being structure breakers. Proline because it does not have a hydrogen to hydrogen bond width on its backbone, and glycine just because it has so much conformational freedom, it doesn't want to be restrained in an alpha helix. So the next type of secondary structure we're going to look at are called beta sheets. And there's two types of beta sheets. You can have parallel or anti-parallel. And the words parallel and anti-parallel go with the direction of the N to C terminus. So what I mean by that is if you look at the top strand of the anti-parallel picture, the N terminus is right here and the C terminus is going that way. So it's going from right to left, while the other strand is going the opposite direction. N terminus here, C terminus. So that's left to right, while the top was right to left. For parallel, the N and C terminus are going in the same direction. Here, they're both going right to left, or they could both be going left to right. What matters is that they're going in the same direction, right? And the way that beta sheets work is shown down here. So when looking at beta sheets, the arrowhead is telling you where the C terminus is, and the flat part of the arrow is the N terminus. So whenever you look at beta sheets in images, they're mostly going to be presented with arrows. Now, in anti-parallel, you usually have a simple loop to connect your strands. And you can see my arrowheads are going in different directions, so that's anti-parallel. While in parallel, you need some kind of connection to loop back around. So my arrowheads are going in the same direction now. And if we think about our sheets being on a plane, as shown here, that's our plane. Our loop has to go above that plane, or it has to go below that plane. Now, when we talk about stability, anti-parallel beta sheets are more stable because their hydrogen bonds are at a 180-degree angle. So compare that with parallel, their hydrogen bonds are at an angle, so they're less stable. So when we look at proteins in nature, we're more likely to find anti-parallel beta sheets than parallel, because anti-parallel are more energetically favorable. Now, the last type of thing to talk about beta sheets is that these connections that connect different sheets, these are called turns. So we have a beta sheet, then we have a beta turn. This turn is what is connecting our different beta sheets. All right. So here we have our first question. Well, we have a chat, so let's take a look at that first. So parallel goes in the same direction with a loop. Yeah, so parallel, you need to like loop over or loop under to make sure that the N and C terminus are running in the same direction. Anti-parallel, you just need a simple loop to continue on with the beta sheet. All right. Go. Yeah. Let me see here. All right. So our first question: which of the following peptides is least likely to form a beta sheet, and why? So I give you three proteins here, or three peptide chains. Which one is least likely to form a beta strand or a beta sheet, and why? And this goes back to what we were talking about on Friday. We have a vote for 2. We have another vote for 2. Question is why? Another vote for 2. Why are we voting 2? What is it about 2? Why not 1? Why not 3? It's, yeah, G and P. It's mostly P for 100%. P, proline, really does not want to be in secondary structure because it can't hydrogen bond in its backbone, right? Proline does not have a hydrogen there. Glycine as well, very flexible, so it really doesn't want to be constrained to the geometry, the fine side ankles of secondary structure. However, we do see proline and glycine in our turns. Why? Proline has a cyclic structure and is able to beta turn. It can do that. What about the cyclic structure of proline that we talked about on Friday makes it unique in comparison to other amino acids? Proline is the only amino acid to be found in blank conformation. P 100% does not form a secondary structure. So a glycine is much more likely to be in a beta sheet or alpha helix than a proline. Proline, like, literally cannot be. Cis. Yes. So proline is the only amino acid that can have a cis conformation. If we look at this term, you see it's very tight. This is like N terminus to C terminus. Most peptides cannot make a like 180, or rather, I guess this is 90, 290-degree turns. Most amino acids cannot make tight turns like this. One advantage of proline is that it can be in a cis conformation, cis backbone conformation, that allows it to be in a turn like this. So proline has this backbone geometry that can be different than every other amino acid. How about glycine? Why is glycine found? The cyclic structure has a lower energy configuration. No, proline is cyclic. That doesn't have to do with energy. What it has to do with, proline can form a cis backbone conformation that no other amino acid can. In that cis conformation, you can obtain geometries that no other amino acids can. That one of those geometries are found in beta turns. So that's why prolines are in beta turns. Glycine is very flexible because it has no steric hindrance, because it's not chiral. It doesn't have to do with chirality at all. It has to do with very little steric hindrance and being very flexible. I I hesitate to use the word no steric hindrance because there is a little bit, but it's very minimal compared to other amino acids. But yeah, flexible, very low steric hindrance. So glycine, and I'm just going to go back a slide. And let me get off my drawings. Clear, please. Okay. If we look at glycine, and we're looking at the Ramachandran map, which is the fine psi angles that the backbone of glycine can take, the only part of the map that glycine cannot take is this dark blue region, right? Every other fine psi combination is available to glycine. Here, in these secondary structures, this is the majority of other amino acids, right? Other amino acids do have some flexibility, but on average, this is what other amino acids can do up here in these regions. So glycine has a lot more flexibility than other amino acids, allowing it to be in these tight turns, which aren't favorable to other amino acids. So glycine, because it's flexible, proline, because it can be in a cis peptide conformation. That's why we can find these amino acids in these tight turns. Questions about that? All right. The last thing we have to talk about, the last couple of things. So we do have a question. What was A again? 2, because there's a proline. Proline cannot be in secondary structures. Okay. So there's 2 main types of proteins in the world. Ones are called globular, where you have hydrophobic amino acids, and we'll talk about this later today. Hydrophobic amino acids make a core, and it makes a cool 3D shape. The other type of protein that we're going to talk about right now are called fibrous or fibrous, depending on how you want to pronounce it. These proteins are basically giant alpha helixes. They're giant helixes, right? That's all they are. And they're often, protective or connective or supportive roles. So what we're doing here is we're looking at one type of protein called alpha keratin. Alpha keratin can be found in your skin, your horns, your nails. Those of you who have feathers, that's where alpha keratin is. Your jello, that's what, jello is mainly collagen, but yeah, it's the same idea. And what alpha keratin is that you have 2 proteins that are wrapping around each other. And each protein forms this pseudo repeat, in that it repeats the 7 similar type of residues, where residue A and D are hydrophobic, while B, C, E, G, F are all hydrophilic. And what happens is that when 2 of these chains come together, these hydrophobic regions want to interact, and these 2 chains wrap around each other. And that's why they're good connective tissue. It's almost like a rope, right? 2 strands of a rope coming together to make a stronger rope. So the question is, I just told you why positions A and D are hydrophobic. Why are the other positions polar or charged then? What is the logic of the other positions being charged or polar? Whoa. To hydrogen bond? No, well, kind of. Hydrogen bond to what? The hydrogen bond to interact with the rest of the body. Yeah, kind of. What are we hydrogen bonding to? Sorry, I said no because I thought you meant hydrogen bonding to itself, but interact with the cell. No, sorry. Then that is not it. To rest, I'll accept like to the rest of the body. So these things are connective. So you're going to want to interact with other proteins as possible. You're going to interact with water. Basically, you're going to interact with everything else, right? And so you want to be polar so you can interact with water or you can interact with proteins. And if you have two nonpolar positions, right, at A and D, these will be forced together because of the hydrophobic effect when you're making this protein, right? So you're going to be making this protein and you want this protein to come together. So the A and D amino acids will glob onto each other, while all the other amino acids will make sure that we stay, we can still be dissolved in the cell when we're being made. And then when we're out in the world, we don't, if the other parts were hydrophobic as well, this protein might come unwound or it might glob to other hydrophobic amino acids. We don't want that. We want specific interactions. So that's generally why it's polar and charge. Basically interact with water, other proteins. Stay dissolved when being made. More or less, that's the reason why. Caw, yes. All right. So if I have two sequences in B, one goes isoleucine, glutamine, glutamic acid, valine, glutamic acid, arginine, and aspartic acid versus tryptophan, glutamine, glutamic acid, tyrosine, glutamic acid, arginine, and asp. Which one will more likely to be a coil-coil? Non-polars to keep the strands together, yes. So polar ones interact with water, other proteins, anything that's polar. The non-polar ones make it stick together because of the hydrophobic effect. Yep, that's the general idea. So for B, which two sequences will form, will form this alpha keratin better? Sequence number 1 or sequence number 2? Who knows their amino acids? Who can look at the 3-letter code and determine what amino acid it is? I can't sing. That's why I teach science. Yeah, exactly. So polypeptide number 1 is way more likely to form this alpha keratin structure because you remember I told you position A, so isoleucine, and position D, valine, they have to come together to have a hydrophobic interaction, and they have to make this close contact. So you don't want your hydrophobic regions to be a huge tryptophan, which is double ring, and huge tyrosine, which is a ring with an OH. That's going to leave you bigger gaps, and it's going to be harder to kind of force those in. So really, your isoleucine and your valine have more of a chance to come together than your tryptophan and your tyrosine. All has to do with the size of our amino acids. Questions about that idea? So the large cyclic rings won't let it. It's more like they're interacting through non-covalent interactions, so they don't bond. But if it's big and bulky, You generally don't want like huge aromatic groups, right? So if you're big and bulky, like my fists, and you're trying to come together, you might have a harder time coming together. While if you're like tiny, like my fingers, probably have an easier time coming together. Because remember, this is all non-covalent interactions. So if they're rings, the only way they can interact is through stacking. While if you're like, this is going to look weird, I know it. If you're a valine and you're an isoleucine, you kind of fit in perfectly. No innuendos there. That's amino acids, by the way, so no dirty thoughts. These are rings, so they have to stack like that. Well, if you have a bunch of rings coming together, they're either going to stack perfectly or they're going to crash into each other and not figure out how to stack. So generally in the middle of two tight coils, this is going to have a much harder time to interact than like tiny carbons. So you don't want big bulky groups. No, it's all hydrophobic interactions. Just like DNA, the rings stack. And the rings, I mean hydrogen bond too, sorry. That's a bad example. These don't hydrogen bond, they have to stack. That's the only way like a tryptophan and a tyrosine would interact, is that's how they interact. They pi stack. That's how they interact. Yeah, I mean, imagine you're staring down this, or you're staring down this, right? So what's happening is that you're taking this and you're flipping it, so you're looking like down where the two things connect. So it's just DA, DA, DA all the way down through there. And DNA are interacting. Well, if you have two giant rings, they're just going to be butting their sides against each other. That's no interaction at all. The way that rings interact is through pi stacking. They have to be stacked on top of each other like a sandwich. That's how all carbon rings will interact with each other. Like benzene, right? If you have a solution of benzene, they're not interacting by just having the rings next to each other. They're interacting by having the rings in layers, so you're gonna have this pi interaction. That's how a benzene solution works. It's the same idea of these big bulky aromatic groups as well. I can't talk anymore. Pi stacking. The ring stack. That's how they interact. It's hard to stack rings in a tight little space like that. All right. The last thing we're going to talk about is collagen. Collagen. No. I was saying that it's harder. The only way for rings to interact are through stacking in a tight little space between the different helixes. It's going to be very hard for them to stack efficiently because of steric interactions. They're going to be hitting each other. So that's why it's less likely they will interact. While something like a valine and an isoleucine are tiny, so they can interact without causing steric hindrances. That's what I was saying. All right, so moving on. So let's talk about collagen now. Collagen is what makes up Jell-O. So anyone who likes Jell-O, what you're basically eating is horse bone. I don't know if they still make it out of horse bone, but they used to. It's very strong, insoluble fiber. That's going to make up your teeth, your bone, your cartilage, your tendons, fibrous materials of the skin, and your blood vessels. Yeah, so next time you eat Jell-O, just think, yeah, look up Wikipedia gelatin, and they'll make you think twice about eating Jell-O. Gelatin's gross. Tastes good though. Okay, so what collagen is made out of is a triple helix. So alpha keratin was a double helix where you have two proteins coiling around each other. Here you have 3 proteins coiling around each other in a triple helix. Now, the amino acids of these triple helixes are relatively simple. It's glycine, then a protein X, or amino acid X and Y. X and Y can be different amino acids, but X is usually proline, Y is usually hydroxyproline or hydroxyprolol. The only difference between hip and pro is that hip gets an OH attached to it, right? Now, for hydroxyproline, the way you make it is vitamin C. And so if you don't have vitamin C, you get scurvy. So 10% of college students, at least in 1999 at the University of Arizona, had mild scurvy because college students eat like garbage. Eat your fruits. If you don't, you're going to get scurvy. Pizza's not a fruit. So what happens is that you can't make collagen. And if you can't make collagen, your teeth start to fall out. Your skin gets all saggy. You can't make good blood vessels. So make sure you get your vitamin C. Since we're talking about this, vitamin C does nothing for your immune system. Stop taking vitamin C at the airport. It does nothing. The person who made that is a giant liar. And it's been in our consciousness for 60 years now. It's proven vitamin C does nothing for your immune system. So stop buying it for that. All right, so that's collagen. Tomatoes and pizza sauce is a fruit. Okay. I mean, chocolate also has antioxidants, but you don't eat chocolate to get a bunch of antioxidants. Emergent C does absolutely nothing for the flu, right? Sorry to burst your bubble. You're buying expensive orange fizzy drink and a placebo effect, I guess. So that's collagen. Next, we have keratin. Keratin is an interesting protein because it is 2 proteins, again, wrapped around each other. So purple and green form what's called a coiled structure. It's called coiled because each individual protein is coiled and they are coiled around each other. Coiled. And the way this works is that a dimer will interact with other dimers to make what's called a protofilament. So 2 strands of dimers coming together is called a protofilament. These protofilaments will go and stack 2 at a time to make microfibrils. And these microfibrils, these keratins, are stuff that make up your skin, your nails, some in your hair as well. So it's just these fibrous proteins that are making our connective tissue, making our hair tissue, making our nails, making our bones. And they're basically just coils wrapped up on each other. Yeah, people who take vitamin C to make them immune to the coronavirus are not scientifically accurate. I was going to say something else, but that's not nice. So I've heard people, so many things about the coronavirus. I'm going to take zinc and vitamin C. Congratulations, you're going to get the coronavirus. It does nothing to help you. What does vitamin C do? Vitamin C makes hydroxyproline so you don't get scurvy. It might do other things. I'm not a vitamin C expert, but I know it does not help your immune system. So how do you boost your immune system? You boost it by eating balanced meals, exercising, and sleeping 8 hours a day. That helps. Right? Okay. So no supplements to help. Honestly, if you want my opinion on supplements, I mean, people might disagree with me. I think supplements, for the most part, unless they're prescribed by your doctor, are a way to give yourself expensive pee, because most of the vitamins and minerals you get that are effective, you need to eat from food, because they naturally come with like other helper proteins that make them easier to digest and easier to absorb in your system. When you're buying like a zinc tablet or whatever, 99.9%, maybe not that high, but the majority of it you cannot absorb. And so what happens is that it just goes to your bloodstream, then you pee or poop it out, and congratulations, You just spend a bunch of money to make the water system have more zinc in it, right? Because we can't absorb high levels of these minerals by themselves. For example, like hemoglobin, we need a special form of hemoglobin that's found in animals to properly absorb that and break it down to get the iron. If you eat like an iron pill, it doesn't have that form of hemoglobin, so it's much harder for your body to break it down. So I'm sorry to, unless your doctors, now I'm not saying all supplements will never help. There are situations where if you are chronically low in something like calcium and your doctor says, you know, your levels of calcium are dangerously low, you need to take a calcium pill. Now, you're not going to ingest most of that calcium pill, but you'll get more than you were getting. And so that will drastically help you, right? So I would say my advice is, unless directed by a physician, don't willy-nilly buy supplements, because a lot of the supplement companies take a little portion of scientific data out there and blow it way out of what their original researchers say. And they will make advertisements based on this little piece of fact that has a bunch of conditions to go with it. So what if you're iron deficient? Yeah, so if you're iron deficient, then you'll probably have to take iron pills because you'll at least get some through those pills. Again, I'm not saying supplements don't work at all. I'm just saying that they don't work that well compared to eating. Like if you're iron deficient, the best way to get iron is to eat like meat, right? If you're a vegan, well, then you have to do this other side. But yeah. Yeah, so the iron deficient doctors normally order an IV iron infuser for faster results. Huh, did not know that. Anyways. So it's a placebo effect, not melatonin, that's working for me. Probably. Like just eating melatonin, I don't think it's going to work all that well. It might have a little bit of benefit, but I would say don't trust advertisement on the side of a bottle, right? Like alkaline water. It's good for your skin. No, it's not. It's not going to do anything. I'm getting way off topic, even though this is fun to talk about and rant about. So gelatin, mostly collagen. Why is it not good? Because it's made out of 3 amino acids, right? So question A is just for you to know what collagen is made out of. B, here's a thinker though. Why is it absolutely required to have glycine in collagen? Because if you look at collagen, it's glycine, amino acid, amino acid. X and Y are usually something, but why do we always need glycine there? Little steric hindrance. Flexibility, they're both like the answer, right? Because you have 3 coils that are packed in each other. So there's a lot of steric hindrance there. So you need your amino acid, who has the side chain of a hydrogen, to really pack in there. And you're making these tight turns, right? We talked about in beta turns, how glycine has so much flexibility compared to all the amino acids. It can adopt conformation that other amino acids can't. So having these tight turns and low steric hindrances allow us to form this triple helix protein that no other amino acid can really do it. So glycine is really specialized for having little steric hindrance and great flexibility. All right, and that's the end of this PowerPoint. What was A? A is because collagen is made out of glycine, proline, and proline. So you need a lot more amino acids than that in your diet. We can make like, I don't know how many. I think we can make like 12 amino acids, and then we need to eat 8 other ones. Glycine, proline, we can make. Proline and hydroxyproline. So if your only protein source is gelatin, 1, I question your life decisions. 2, you're not going to get the amino acids you need to make. But the answer to A is up here because it's made out of 3 amino acids. All right. So let's move on to our new PowerPoint. And by the way, let me just say, because my wife sometimes yells at me when I go on rants like that, I am not a medical doctor. I'm just a biochemist. So keep that in mind. I'm not a medical doctor. I'm not a nutritionist. I can just tell you the biochemical side of stuff. All right, let me share this. Current slideshow. All right. So we talked about primary structure, and let's just go through a little reminder here. Primary structure is just the amino acids N-terminus to C-terminus. Secondary structure is the local backbone conformations of your protein, so your alpha helixes and beta sheets. We are now on tertiary structure. What is the overall 3D conformation of one peptide chain? All right. What is the overall 3D conformation of just one peptide chain? That is tertiary structure. And what we're going to talk about is the basics for how to find out tertiary structure. How do we know what proteins look like? That's what we're going to talk about. The first technique that we use is called X-ray crystallography. And what you do is that you take protein at high concentration and you make a crystal. So what you're seeing here is a protein that was made at very high concentration to eventually form a perfect crystal. Now, those of you who like art and like trial and error, you would love protein crystallography, because what you do is you set up hundreds to thousands of different conditions where you have different salts, different concentrations of salts, different solvents, different types of solvents, different temperatures. And what you're doing is you're trying to get a nice, perfect crystal made out of protein. Then what you do is you take this crystal and you shoot it at an X-ray while spinning the crystal at 360 degrees. The high-level version of this is done at synchrotrons. There's one in Chicago I know of. Since I'm from the Midwest, that's the one I always think of. What a synchrotron is that it's a giant facility. And I'm talking about like a quarter mile to a mile giant circle. And in this circle, what you do is you use magnets to shoot electrons in a circle, making them go faster and faster and faster. And then in the other direction, you can shoot another beam of electrons or atoms and have them crash into each other. This is how we do high-level physics, how we find quarks and gluons and make new elements. Well, while the physicists are doing that, the benefit of having electrons travel very fast in a circle is that at right angles from the circle, X-rays are being shot off from the synchrotron. So you have X-rays coming off of these beams. And biochemists sit in little rooms and they harness these X-rays to shoot crystals, right? So that's how it's done in the real world. If you know that X-ray crystallography is done by taking a crystal and shooting it with an X-ray, we're good on that front. Okay. Now, what do you do when you shoot an X-ray with a crystal? Okay. So what will happen is that the X-ray will hit the nuclei of your atom and they will bounce. And you'll record this bounce on a piece of more or less film. And so each dot here is one of your nuclei, one of your atoms bouncing, one of your amino acids getting hit by an X-ray and bouncing that X-ray and making that signal. Then you take that signal and you make a 3D map So you read this in and you get this 3D map out. If this was a biophysics class, I would spend the next week and a half telling you how we convert this to a 3D map, and we would do actual problems where we would solve the math for that. In biochemistry, we can just say computers. Computers do this for us. They take this 2D image and make a cool 3D image. So no need for high-level physics here. And then with this electron density, you fit your atoms to the grid. So each density just tells you, there was basically a nucleus here. I have electrons around here. Then it's your job to fit your amino acids into this grid. And so what happens is that a computer, again, our friend will do the first pass, and then you as a human will refine the structure. And your goal is to get the highest score possible. It's basically like a video game where you try to fit your structure into this grid, and the better you do, the higher score you get. Those of you who want to try this, look at Fold It at Home. It's out of like Washington, I think. And basically they have you as the player trying to solve real life protein structures. And what's happened is that they've been able to publish real scientific papers by using not scientists to do this. Basically, you just have gamers follow a set of rules to a bunch of biochemistry physics rules. They solve a paper, and there's one paper with 4,000 people on it because all the users, and it was just their usernames. It was actually pretty funny. But yeah, check that out if you want. Yeah, it should be Fold It at Home. I believe that's what it is. And so I have a question here. I'll just answer it because we're running a little long. So when you get an X-ray structure, what usually happens, if this is my peptide, usually you cannot see the N and C termini. Usually they never appear in your structure. And the reason why is that the N and C termini are very flexible. To have diffraction, and that's what this is called, to have proper diffraction, in your protein crystal, you need the protein to be repeated perfectly over and over and over again in your crystal lattice. That's how we can see any signal. Basically, every time, I know I was going to talk about the physics, but I'm going to talk about the physics. You shoot your X-ray, and it comes out at an angle. And if you have a protein that's perfectly in line, just a little farther back, the angle will be the same. And basically what happens is that all these angles amplify each other. All these signals amplify each other, and you get a dot. Well, the N and C terminus of like any protein are flexible, and so they are not in the same position. Every single protein will have a slightly different position, and instead of amplifying the signal, they destroy the signal. So the answer is N and C terminus are flexible. In a crystal, you need to have the same geometry for this to work, i.e. no movement, no different positions. The position has to be the same. So any questions about X-ray crystallography? Those of you who are interested in physics as long with biochemistry, seriously give biophysics a look. That's the field I was in. If you like to play with computers, maybe you're crap in the lab like I was, but you still like this stuff, biophysics is your friend. So just the N-terminus at the beginning and C-terminus at the very end. Those aren't the only ones. It's possible other part of your proteins are also flexible, but those are the ones that are the most common. So it's the easiest way to make a broad statement. So all we need to know is X-rays determine tertiary structure, and you shoot protein crystals with X-rays to get an image. And that idea about the N and C terminus being flexible, and that's why you don't get an image. But otherwise, yeah, that's basically the Cliff Notes version of this whole slide. I could just say that, but then you wouldn't get all this cool information. Any other questions? If you can't tell, I really like biophysics. Out of all the biochemistry, structures is my bread and butter. It's my jam. All right. The second way to do this, your friend from organic chemistry is NMR. So I know with Shelton, I believe, those of you who took organic chemistry with Shelton here at Tamusa, I believe I've seen this, I've seen people cry over this. You have to like solve an NMR spectra, right? And you get a couple peaks and it's like, what is this? Well, if you thought that was bad, here's a common spectra you get from biochemistry. And as a grad student, I had to solve part of that. So for biochemistry, our NMR is a little more advanced than organic chemistry. We do multiple nuclei, right? We can do hydrogen NMR, carbon NMR, nitrogen, phosphorus NMR. And basically what you do is you do different types of experiments. And the way that NMR works is that you shoot it with this magnetic wave. Based on the environment of your nuclei, they will absorb these waves at different frequencies. When they absorb these waves, you get a readout. And that's the readout of biochemistry, just one experiment. Why would you do that to yourself? I didn't do it. My instructor did it because the only way you know how this works is by doing these calculations by hand. Of course, a computer does this, but if you don't know how it's done by hand, then you don't know how it's done, right? So there's two different types of NMR that's commonly done. One's called a cozy, and basically you measure the distance between hydrogens, so this distance that I just drew, when they're connected to one or two other atoms. So every single hydrogen in your structure, you'll measure a cozy for. And then you'll also measure a nosy, where you'll say atoms that are close. How far are they? So you might do that with a carbon, nitrogen. And you get like thousands of distances, right? And along with other constants, such as bond distances, angles, planarity of rings, chirality, van der Waals, you put all this together. So you're trying to solve a puzzle, but there's like 100,000 rules with these puzzles. And because of that, you get structures by following these rules. I guess I should use a different pen. Black doesn't show up so well. You get a bunch of different structures that follow these rules that you set. Now, my question down here is cut off, but I'll ask it. In X-ray crystallography, you get one structure, like 95% of the time you only get one structure. The other 5% of the time you might get two structures that differ a little bit. In NMR, you get what's called an ensemble, which you usually look at like 10 structures, 15 structures, 20 structures. So why in NMR do you get multiple structures? While in X-ray crystallography, you only get one structure. And after this question, we'll call it a day. because the X-ray is static, so the structures don't change due to the movement. Yeah, so both of those are, so we have the structure doesn't change in X-ray while there's movement in NMR. Yeah, so hopefully you remember from organic chemistry, when you do NMR, you're not in a crystal, right? You're in a solution. When proteins are in a solution, they do what we call breathe. They might have a fixed 3D conformation, but you have movements, like they're breathing in and out because they can naturally move. So it's almost like the proteins are breathing. Each one of those breaths can be captured using our NMR, where we get slightly different rules depending on what breath we're looking at. And since we have thousands of copies of proteins, we get a bunch of snapshots. And when we try to solve our puzzle, we get a bunch of solutions. So here, let's say this is phenylalanine. Each one of these little blue lines is a different solution where the phenylalanine could have been. Here's obviously tryptophan. Here's tyrosine, right? Each one of these images we're looking at kind of looks almost blurred because there are a lot of different positions that tryptophan could have been at. Notice that all the lines are close together, so it's not like drastically different. It's just a little different because NMR is done in solution, so you get to see the breathing of the protein. Crystallography is done in the crystals, so the proteins have been Han Solo'd. If you ever seen Star Wars, where he gets frozen in the carbonite and he's stuck, that's basically an X-ray crystal, except Han Solo would be copied 1,000,000 times in each direction. That's the difference between NMR and X-ray crystallography. That's the big idea I also want you to get away from, like, what's the difference between NMR and X-ray? How do we solve them? And all that good stuff. So every time it moves, you get a picture and you stack those pictures to get mini structures. Yeah, more or less, because you're doing this experiment. So your nosies, your NMR is going to be a little messy. It's not going to be nice straight lines like you'd see in organic chemistry. Your signals are more spread out because you have a lot of different proteins adopting slightly different positions. And by the spreading out of this structure of these signals, you have more solutions. And so you solve those multiple solutions. So generally, when you do NMR spectroscopy, can't talk anymore. When you do NMR, you usually get the top 10 structures. And like I said, they don't look very different, but you just present, these are my 10 structures that were most representative. And this is what the protein looks like. And generally, they look the same though. Any other questions? If not, I'll put up Friday's lecture. Sorry, I forgot about that. If you're ever looking for a lecture that we did previously and you don't see that up, just shoot me an email to have remind me about that. I was a little bit busy last week, so it totally slipped my mind. But I'll do that. I'll put up today's lecture, put up a homework as always. Other than that, we're done. Don't take vitamin C to get over the flu. Doesn't help. Otherwise, hope you all have a great day. Oh yeah, vitamin C does not help with COVID either, so don't believe that. All right, bye.